happy with that?
Yeah, I thought I was in the next session. Okay, thanks. So I'm standing in for Irina, and Irina also prepared the slides and explained to me what she wanted to say. Irina can't be here. She had a small accident, but everything's fine, but she just can't be here. So the talk is about network-based predictive models. And I think basically, I don't have to go into detail, but we are dealing with fMRI data. We have four-dimensional images, like basically a series of 3D data acquired in the brain that shows us something about the bold signal. And the challenge is now, how can we predict from these signals what will happen to a patient, or what is kind of the diagnosis of the patient, or how can we relate, or what in the signal do we actually want to relate to clinical findings, or to outcome, or to anything that we would like to know from the data which we don't a priori. And I think we saw some very nice talks about that right now. And in this talk, I'd like to, or Rina wanted to focus on network-based models. So why network-based models? Well, we need some predictive features, some biomarkers if we want to feed our predictive models, and these features can be either voxel-based, or activations, or patterns of activations in different voxels, or they can be really network-based. And one of very clear-cut motivations for doing it in a network-based world is because the brain is a network. And sometimes you can argue that, well, what really matters is the properties of this network, and not so much what's happening at an individual voxel. And so there are two kinds of networks this talk will consider, and this talk will be about. So one is just functional networks, or the thing functional networks is usually, or the term is usually used for, which is basically just big correlation matrices that describe the signal behavior observed in fMRI data. The second kind of networks is Markov networks, which is a bit very closely related, which is basically based on the precision matrix, or the inverse covariance matrix. The difference here is that the covariance matrix is basically showing you the marginals, so the overall covariance structure. The precision matrix, so the Markov models, are dealing with the actual conditional correspondences. So it's a much sparser relationship family, so there are a lot less conditional dependencies usually compared to the overall covariance occurrences. And well, why networks? I think that's, if you're in a multivariate pattern analysis course, everybody is kind of agreeing on that the multivariate characteristics of the brain are relevant, and networks are just one very powerful way of looking at these multivariate characteristics, and specifically looking at the internal architecture of these multivariate characteristics. All right. So there is a lot of literature on why networks are important, and on different approaches towards networks. I'd like to start with an example where it shows how networks are sometimes much more informative than individual voxels, and especially in the one example of schizophrenia, which is also considered to be a network disease. So here we have a standard fMRI experiments. People are hearing a sentence and they have to give a response, and that's on and off sequenced standard block design. And we have a small patient group of schizophrenic patients which are actually prone to auditory hallucinations, and we have a normal control group of 11 other patients, other subjects. We do a standard experiment, and then we compare voxel-wise if there is a difference in the response to the stimuli and the actual task performance that differs between the two cohorts, so between the patients, the schizophrenic patients, and the normal controls. And it turns out that when we do that in a very standard way, there is no statistical significance that we can kind of grab from the data. If we do multiple comparison correction, everything vanishes, and we turn up with data where we don't see any difference between these two cohorts. Now what we can also do is to look at the networks, which is fairly simple once you have the individual voxel values. You just do standard correlation analysis, so you calculate basically the correlation among all of these individual signals, and this ends up with this matrix in the second line, and you can threshold that and say like even further simplify it by just thresholding it and say, well, everything below the threshold I just forget, and everything above the threshold, that's a link. So that's a link based on the correlation strength. And then you have a graph among all the voxels. Now you can look at something like the degree map of these voxels. So the degree map would be just how many connections are connected to a specific node. So again, you have a feature that is assigned to each voxel, but this time the feature is not based on the individual voxel signal, but on the signals of other voxels in relation to this one voxel. It's basically the connections to this one voxel. And it turns out that when you do that and compare this, again, voxel-based feature, but a feature that expresses network characteristics, you do get significant differences between the cohorts. And they are actually meaningful for neuroscientists that do study this disease. So on the map down here, that looks similar to a standard activation map, but this time it's difference in the degree maps between the schizophrenic patients and the normal controls. And the interesting thing here, or the one that makes it plausible, or that is a nice result about this, is that it's actually in the auditory cortex, where you would expect these differences to be for people who have auditory hallucinations. Okay, so these are just to further go into detail about how these features are actually stable and repeatable. And it turns out that these degree-based, network-based features are also very, very stable, also in comparison to individual network voxel activations, because some of the regularization is just built into this network level analysis. So in this graph, what's shown is that in repeated experiments, the number of top-ranked voxels, so that's the x-axis. When you look at the overlap of these top-ranked voxels among different trials, you see a very high overlap between different trials in these network-based features. So it's a very reliable source. It's not just noise in the data. Okay, so they outperform a standard classifier on voxel-based features. There's some more work where you can go beyond individual voxel-based features and say, well, I'm now actually interested in the links between pairs of voxels. So I'm just treating, instead of the entire voxel space and features that can be assigned to individual voxels, I treat all possible edges between all possible voxels as potential features. And then, of course, you immediately have to go for something that reduces this number and for feature selection in order to turn out a classifier that relies on a subsampling, on a subset of the possible edges in the graph. And so this link structure is also a very nice predictor for certain classification questions. But I think the key point that Irina wants to make and that her group's work is about is that now that you're in this space of edges in the graphs, well, on the one hand side, you have a problem about reduction, about how to choose the edges and that are actually predictive or that you use for the prediction, how to render this meaningful, this selection, and that this problem is much bigger than just selecting the voxel. So if a couple of thousand voxels gives you many more edges you have to select from. And so this selection process is something you have to think about, because it's not only individual features you're selecting, but you're selecting links. And I'll just come to why this is important and how you can view this problem. So first of all, we get network features as schizophrenia markers. So we can look at the voxel degrees, we can look at the link weights, at the voxel strengths, absolute strength. So these are all different ways of extracting voxel level features or link level features from graphs. And there is wide literature. So there is, for example, one quite popular paper by Bullmore and his colleagues in NeuroImaging, which describes these measures, and in many of the toolboxes it's just built in. So if you have your signals, you can extract the features and can use them and try them out for your classifiers. Okay. One thing though is, well, you have a graph, you can either build it based just on a covariance structure, just on the correlation structure, or you can do one step more and say, well, all this correlation structure is actually the result, the picture I get, because there is some underlying conditional structure in the graph. And Markov network or Markov random fields is a family of approaches that tries to tackle this problem of figuring out what are the conditional actual connections within this graph that cause the signal correlation pattern across the entire graph. So the basic assumption is that I don't need all the connections actually being there to create a certain correlation pattern, but I only need a subset of actual conditional connections between points to create these correlation patterns, because correlation can be also built based on some path that jumps along a couple of edges. And so what this is about is finding this underlying edge structure that creates the signal, even if it's much sparser than the initial covariance matrix. And just to remain in this Gaussian framework, the Gaussian Markov random fields, the key idea again is that we have a subset of links, and only this link are actually meaningful, and all the other ones that are not meaningful are just not there. So that's the Markov property. Okay. I'm just going through this, but I think that's more, the details are more important for reading up on it later. All right, and so now we get the problem of how do we get these conditional connections between individual nodes if the only thing we can actually observe is the covariance structure. Because the covariance structure is pretty easy to get, we just calculate the individual covariance values among different voxels. The more difficult thing is what's the underlying conditional structure? How can we estimate this conditional structure? And so it turns out the most easy way is just to invert the covariance matrix. But inverting the covariance matrix in the scenario we have here, where we have very few samples compared to the overall number of voxels, so you have, no matter what experiment or no matter how persuasive you are to keep people lying in the scanner for a long time, you will never get as many time points as you have voxels in your data, or very seldomly. And so inverting this covariance matrix becomes difficult and very unstable. And so it's not just about inverting it, but it's really finding ways to regularizing this inversion process. And I won't go into detail here, but the key insight is that there is an entire community of people, mostly presenting at NIPS every year. So every year there are a couple of new algorithms dealing with this problem of inverting this matrix and in finding a stable estimate for the underlying conditional structure that is actually generating your observation. But the key insight for us, I think, is that these edges are very valuable because they tell you a bit more about the connectivity structure in the brain than just looking at the covariance matrix. So here are just a few approaches that try to do that. And the one thing, or the last thing, or I think one of the key points in the talk is that one approach by Jean Honorio at AI Stats is that when you think about this problem of variable selection, most of the regularizers that are applied during this inversion of the matrix actually aim at sparsifying the problem, of getting rid of as much as possible, so setting as many of the variables to zero and keeping a few of the variables that are actually predictive. So that's very similar to Gael's talk and to many other approaches where our basic assumption is that the contribution to our prediction is not a little bit of everybody, but it's just a few main predictors that are in our data. And our task is first of all to predict, but also to actually find out what are the informative variables. And now if you think about networks, you can just treat the individual nodes as individual potential variables and to enforce sparsity there. Or you can do it by the edges and say, well, I want a sparse subset of edges because I believe only a subset of edges will be relevant for my prediction task. Now, just using a sparse subset of edges is problematic because there is much more in the architecture of your predictors than just a list of independent edges. All these edges connect to nodes. And so what this approach is about is connecting these two sparsification criteria. So what they did basically, Sean and Irina and their colleagues, just clicking so that you see the entire slide, was experimenting with different cost terms that ensure that when you select a sparse subset of variables, a sparse subset of edges, that not only the edges will be sparse, so not only will you get a subset of edges, but this subset of edges will also connect a subset of nodes, which means you will prefer the nodes which have a lot of edges connecting to them, and will discard edges and nodes where only a few sparse edges are flying around somewhere in the brain, connecting some node somewhere. Basically, it pushes for sparsity in the edges, but it also pushes for edges to be selected that connect nodes with high degree, very coarsely speaking. And so one example was again based on a dataset by Rita Goldstein. So this is research into cocaine addiction and what cocaine addiction, how this correlates to the reward recognition processing in the brain. So they had in 15 cocaine addicted subjects, 11 control subjects. In terms of prediction, this performs roughly the same. So selecting just sparsity in the edges versus sparsity, group sparsity, selecting both nodes and edges at the same time. But in terms of what the final selected edge sets are, there is a big difference. Before I come to that, I'd like to point out that is subsample data. So you see here in the second line, it's 4 by 4 by 4 cubes, because there's a certain limit right now still on these approaches in terms of how many variables they can actually work with. So in the order of 900 variables in this case, and then it works. Or not 900 variables, 900 nodes, so much more variables. Okay, now the interesting thing is, I think that's one of the most important slides in the talk. So both of these are predictive edges with regards to cocaine addiction versus control. Both of them were selected with a sparsity criterion, so both of them were pushed towards a low number of edges that are used as predictors. But in the lower one, that was the only criterion, low number of edges to be predictive. In the upper one, it was low number of edges plus edges that are connected to high degree nodes, as enforced by this cost term. And it turns out that, first of all, both of them have approximately the same prediction performance, but the upper one is much easier to interpret and actually connects or shows connections between regions that make sense also in the neuroscientific terms. Here are just some of the links or speculations at this point about how this links to insights that are known from neuroscience. So we got connections between visual cortex, prefrontal cortex, and there is, well, a decreased density of interactions between the visual cortex and other brain areas. So in the cocaine addicts, this prefrontal cortex, the role of the prefrontal cortex in the processing becomes different to controls. And that's consistent, at least with some of the published literature, like, for example, Lee. All right, and so with this, I'm at the summary, and Irina asked me to promote 2 books of hers, which I think are really very interesting and worth promoting. I'm just clicking that you see everything. So in summary, first of all, using network measures as a means, as a basis for prediction makes a lot of sense, because it seems that for many of the diseases that are interesting in a neuropsychological sense or in the psychology that affect high-level cognitive areas, the network properties are actually meaningful and involved as a correlate to these diseases. We can do that. There are lots of approaches how to approach networks, how to use network measures as predictors. And it opens an interesting field of how to enforce sparsity. So very similar to enforcing sparsity in the seed voxels and the voxels that are used as predictors in standard MVPA, we can do the same thing in links in networks. But if we do it in links with networks and we want to still end up with interpretable results, we have to think a bit more about how essentially invert the covariance matrix to get a precision matrix that tells us about the conditional relationships. And it turns out that by kind of combining sparsity in the nodes and sparsity in the edges, roughly speaking, we get interpretable results that yield pretty good prediction performance, and are therefore, as much as we can do in providing neuroscientists with hypotheses or basis for hypotheses about which brain regions and which connections among brain regions are involved with diseases. And so the two books are, one is about the theory behind sparse modeling, and one more recent one is about the practical applications of sparse modeling by Irina Wish, and so I personally would recommend them. And with that, I thank you for the attention, and thank you for putting up with me as a replacement. And maybe I can answer some questions, but I think Irina is also very happy if you ask her questions directly by email. Thanks.
Well done, joke. Sorry.
Any questions? Or maybe not.
You have a question? Okay.